So hi students, in last video we have seen about the carbohydrates present in your chapter, that is biomolecules. Now we want to know about another important complex organic molecule present in our body or in living system for the growth and development, that is nothing but proteins. Okay, so these proteins are also said to be the nitrogenous organic complex compounds, and they are found in the protoplasm of all living organisms. Okay, and they are found in the protoplasm of all living organisms. These proteins are made up of amino acids. Okay, so amino acids are said to be the building blocks of these proteins. So to make a protein, it requires amino acid. When n number of amino acids combine together by involving some chemical reaction, it can be converted into protein. That's why we can say amino acids are the building blocks of proteins. Okay, so first we'll discuss about what are amino acids, then we will come to know about the proteins. So first we'll see what are amino acids. Amino acids are the bifunctional organic compounds. Why it is bifunctional? Because it has two functional groups. Amine indicates the presence of NH2 functional group. Acid indicates the presence of acidic carboxylic acid functional group, that is COOH. So because of having two different types of functional groups, okay, these are said to be bifunctional organic compounds. Okay, and especially alpha amino acids are said to be the building blocks of almost all the proteins. Okay, there are different types of amino acids based on the position of the carboxyl group with respective to the amine group. Okay, then these are said to be alpha amino acids, beta amino acids, gamma amino acid, delta amino acids, and so on. In that specifically, alpha amino acids are said to be the building blocks of proteins, building blocks of almost all of the proteins. Okay, that means proteins are containing specifically alpha amino acid. Alpha amino acid means with respect to the carboxyl group, if amine group is present at alpha position or on alpha carbon atom, then those are said to be alpha amino acids. Okay, there are 24 natural amino acids are known. Okay, generally these amino acids are indicated by a symbol, that is C, COOH, NH2, H and R. So this is the general representation of amino acids. Here you can observe. So this is the major functional group, which is nothing but carboxylic acid. So carboxylic acid functional group adjacent carbon is called alpha carbon. To this alpha carbon, amine group is attached. Okay, so to the alpha carbon of acid, amine group is attached. That's why we can call these amino acids as alpha amino acid. These are said to be alpha amino acid. So this is amino group and this is carboxyl group. Okay, both amino group and carboxyl group present in the same molecule. That's why we can call them as bifunctional organic compounds. Okay, these total 24 amino acids, there are 24 amino acids, naturally occurring amino acids are there. So these 24 amino acids are mainly classified into two important types. The first one is non-essential amino acids. Non-essential amino acids. Non-essential, we know what is essential. Essential means required. Okay, non-essential means it's not required. Why they are not required? Because human body itself can prepare this type of amino acids. Okay, human body can synthesize 10 amino acids and hence they are called as non-essential amino acids. They are available in the human body itself. Then why we need to take by external food? Okay, so that means they are not essential to take through the diet because they are synthesized in the human body itself. That's why such type of amino acids are said to be non-essential amino acids. Okay, these are synthesized in the body, need not to take through the diet. So example, there are totally 10 non-essential amino acids. Okay, next essential amino acids we will see. Essential means they are required, need, okay, because they are not synthesized in the human body. These type of amino acids are not synthesized in the human body but have to be taken through the diet. But we have to take them through the diet. Then such type of amino acids are said to be essential amino acids. Okay, amino acids which the human body cannot be synthesized and are supplied in the human diet are called essential amino acid. Deficiency of these amino acids can cause different types of diseases. Okay, so whenever these essential amino acids are deficient in the human body, then they leads to cause different types of diseases. So that we want to take optimum amount of these essential amino acids through the diet. Okay, and remember one more point, all these amino acids, whether it is essential amino acid or non-essential amino acids, they can exist as Zwitter ion. Okay, they can exist as Zwitter ion. Zwitter ion is nothing but the dipolar ion structure is commonly known as Zwitter ion. Okay, at certain pH, the amino acids can exist as a dipolar ion. Such dipolar ion is called Zwitter ion. Okay, it is a dipolar ion of amino acid. Such dipolar ion is called Zwitter ion. Example we can see here are CH, COOH and NH2. This is general representation what I have given here. I took that. Okay, when it involves in, here you can see in this general formula of amino acid contain both acid as well as amine. Amine is having the property to attract a proton due to the presence of lone pair of electrons and nitrogen, and acid is having the property to lose proton, hydrogen as a proton. Then whatever the hydrogen present here, that can be eliminated as H+. That H+ is accepted by the NH2. Okay, it is accepted by the lone pair of electrons present on nitrogen. Then it can be converted into RCH, N, H2 already there, by adding one more hydrogen it becomes NH3. Due to losing electrons it will be having positive charge. Then here COO minus, because H+ is removed. Okay, here two charges containing ion is obtained. This is called dipolar ion. This dipolar ion is commonly known as Zwitter ion. Okay, in acidic medium amino acids exist as cation, while in alkaline medium they have negative ion. Okay, the pH which an amino acid does not migrate towards electrode is called isoelectric point. Now we want to know one more important thing related to amino acid, that is isoelectric point. Okay, this is also another important property of amino acid. So till here I think you are clear. Zwitter ion is nothing but the pH at which the amino acid can exist as a dipolar ion. These two are the two poles, dipolar ion, then that is called as Zwitter ion. Now we will discuss about what is this isoelectric point. So this isoelectric point is indicated by the symbol Pi, isoelectric point. So the pH at which an amino acid does not migrate towards any electrode. Okay, neither it attracts towards cathode nor towards the anode, then such type of pH is called isoelectric point. Okay, that means there is no movement because there is no charge formation at that pH, then that pH is called isoelectric point. Different amino acids have different types of isoelectric points. That means amino acid to amino acid, that isoelectric point gets changes. Okay, all amino acids except glycine are optically active. This is another important property. This is first property, existence of Zwitter ion. and it can also show isoelectric point. That means at this pH neither it moves to cathode nor towards the anode, then that point is called isoelectric point. Next property of amino acid is that they can also show optically active or optical activity. They are optically active except glycine. Okay, except glycine, remaining all are optically active, because this glycine is not having a asymmetric carbon. Glycine formula is NH2 CH2 COOH. There is no asymmetric carbon. Due to the absence of asymmetric carbon, glycine do not show optical activity. To show optical activity, at least one asymmetric carbon should be present, but in glycine there is no asymmetric carbon, then it is optically inactive, except that glycine, remaining all are optically active. So these are the important properties of amino acids. Now we will see how the protein is formed. So before going to study about this protein formation, first we want to know what are proteins. So these proteins are said to be nitrogenous complex organic molecules. Okay, these are nitrogenous complex, which is large in structure, organic molecules. Okay, then such type of organic molecules are called as proteins. And these are said to be essential constituents of all living cells. So first we will discuss about biological importance of proteins. Okay, biological importance. That means how they are important towards the living tissues or living organisms. So first important point is that proteins are required for the repairing the wear and tear of the tissues. Okay, for repairing tissues, it is very important. Next, some proteins can act as hormones. Such as insulin, oxytocin, like this, etc. Okay, all these are the proteins which can act as hormones as well. Okay, these proteins can also act as hormones. Example, insulin and oxytocin. Next important biological importance is all enzymes are proteins. Okay, all enzymes are said to be proteins which catalyze the physiological reactions. Almost all enzymes present in the human body, they are proteins. Okay, these enzymes can catalyze physiological reactions. Okay, next, another important use of this proteins is proteins functions as biological structural materials. So they can act as biological structural materials. Example, we will take hair, nails, etc. So all these are acting as biological structural materials. They are examples of proteins. And one more important use of this protein is hemoglobin. The oxygen transfer, which is a red color pigment in the blood, is protein. Okay, hemoglobin is present in the blood. It is a red color pigment. Okay, this is also said to be one of the protein. So for oxygen transfer, we require hemoglobin. So the red color pigment which is present in the human blood is ready to transfer the oxygen from one organ to the other organ. So that hemoglobin is also said to be one of the protein. Okay, next one. Antibodies in the blood which provide resistance to disease are also said to be proteins. So we need antibodies to fight against the different types of diseases. Such antibodies produced in the body is also example of protein. Okay, antibodies. are also said to be examples of proteins. And then nucleoproteins are the molecules of life. They are the important constituents of genes which transmit genetic information or genetic message in cell division. So such type of nucleoproteins or nucleomolecules are also said to be the proteins. Okay, nucleoproteins. Such nucleoproteins can transfer the message from one generation to another generation during cell division. So these are the few important biological importance of these proteins. So because of having all these biological importance, now it's very need to study about these proteins. So first we'll see what is the structure of proteins. Okay, proteins are built up of a large number of alpha amino acids which are interlocked by the elimination of water between amine group and acid group. Okay, so whenever two amino acids come closer, so they involved in the condensation reaction loses one mole of water molecule and can be converted into a protein. Example, I am taking here two different types of amino acids, NH2, CH2, COOH plus NH2, CH, CH3, COOH. This is one amino acid and it is another amino acid. I have taken two amino acids. When these two amino acids involved in the removal of water, dehydration process. So for our convenience, I am writing these two hydrogens separately like this on this nitrogen atom or amine group. When OH group, so on this amino acid, OH is adjacent to the amine of next amino acid. So then this OH takes the hydrogen present on the amine group of next amino acid involved in the removal of H2O. Water is removed. Then C double bond O group is added to the NH and we will get first peptide. First write from here, NH2 single bond, CH2 single bond is attached to CO. Now to this CO, NH will make a bond. NH bond. Beside NH what is present? CH, CH3 and CH beside COOH is present. Okay, now this is the peptide which is obtained. This dipeptide contain one peptide linkage. Okay, so the linkage which is present in this peptide, dipeptide is called peptide linkage. This is peptide linkage. That means in amino acid we can see peptide linkage. So this is obtained from glycine. This is glycine and alanine. This is alanine. Okay, when these two are combined together, it leads to give a protein which is said to be glycyl alanine. Glycyl alanine. So this is called dipeptide. Dipeptide contains only one peptide linkage. Tripeptide contains two peptide linkage. Tetrapeptide. Tetrapeptide is involved by the addition of 4 amino acids, but it contains only 3 peptide linkages. So it can be calculated by using n-1 formula. Okay, n is nothing but number of amino acids. Okay, n-1 formula is used to find out number of peptide linkages. So what is n here? Amino acids. How many amino acids are involved for the formation of this peptide? 2 amino acids. In place of n you can write 2. 2 minus 1, what is the value? 1. So that means the formed dipeptide is containing only one peptide linkage. Okay, that is about the structure of this amino acids. So proteins. The product is the peptide and the grouping present here is nothing but CONH. The CONH is called peptide linkage. Don't forget, which is very important. What is the type of linkage present in the proteins? That is nothing but peptide linkages. So polypeptide. Polypeptide means many. So peptides are classified into di, tri, tetra and so on. So poly means many. If a peptide is containing more than 100 amino acid units and containing higher than the 10,000 unified mass molecular weight, then such type of polypeptide is considered as protein. Smallest peptides do not considered as the protein. So a polypeptide which is containing more than 100 amino acids and a molecular weight which is higher than the 10,000 unified mass, then that is said to be proteins. Okay, so such proteins are classified into again different types. First classification we will discuss. So classification of proteins. Okay, proteins are further classified into different types. Example, simple proteins, conjugated proteins and derived proteins. So the first type of protein is said to be simple proteins, next conjugated proteins, next derived proteins. So simple proteins, they can yield alpha amino acids on hydrolysis. Okay, on hydrolysis they yield only alpha amino acids, no other byproduct is formed. So that such type of proteins are called simple proteins. Next, conjugated proteins. So they produce a mixture of alpha amino acids as well as non-proteinous part on hydrolysis. Whenever they undergo hydrolysis, they can produce alpha amino acid as well as non-proteinous part. That non-proteinous part is also known as prosthetic group. Okay, so these two are obtained whenever such conjugated proteins undergo hydrolysis process. Okay, example here we will take lipoprotein, nucleoproteins, etc. Okay, lipoproteins or lipoproteins and nucleoproteins, etc. can show such type of can be taken as example of conjugated proteins. Next, derived proteins. These derived proteins are the intermediate products of hydrolysis of proteins. They are the intermediate products of hydrolysis of such proteins. They are said to be derived proteins. That is about the classification of proteins. Proteins are mainly classified into three types: simple proteins, conjugated proteins and derived proteins. Now we have to know about the structure of proteins. Okay, structure of proteins. So the main important condensation polymers of alpha amino acids which are connected through the peptide bond is called the main important structure of proteins. In this structure of proteins, n number of amino acids are condensed and they are joined by the peptide linkages. Then those are said to be structures of proteins. There are four important structures of proteins, that is primary, secondary, tertiary and quaternary. Okay, primary structure, secondary, tertiary and next quaternary structure. Okay, these are the four important structures of proteins. Next one, denaturation of proteins. What is meant by denaturation? Denaturation of proteins. So denaturation is nothing but protein found in biological system with a unique three-dimensional structure. Okay, it is containing a three-dimensional unique structure. Tertiary structure is containing such type of three-dimensional structure of protein. So even in the biological system, it is present in that three-dimensional structure and show biological activity. That is called a native protein. Okay, a native protein which is containing three-dimensional structure and can able to show some biological activity, then such type of protein is called native protein. When a protein in a native form is subjected to some physical change, is undergoing some physical change, like whenever you change the temperature or some chemical change is occurred in the pH level, then the hydrogen bonds are disturbed. So some hydrogen bonds are present, such hydrogen bonds can be disturbed. So whenever such hydrogen bonds are disturbed, they are ready to lose their biological activity and that process is called denaturation process. Okay, see, in the human body, the protein is present in the form of native state, that is called native protein. Whenever it is subjected to some change in temperature or heating or by treating with the acid, so or changing the pH level, immediately it loses its biological activity. That means it becomes biologically inactive. and that process is called denaturation process. Here native protein is biologically active in state. So that biologically active state of protein is now converted into biologically inactive state by making it to undergoing some heating or by treating with the acid. Then that process is called this denaturation. So example, coagulation of egg. Okay, egg white on boiling in a common on boiling is just taken as an example of denaturation. Understand? So like that we will take different types of example for this denaturation. Denaturation is nothing but conversion of a native protein or biologically active protein into biologically inactive state by treating it with an acid or heating. Then that type of process is called denaturation of protein. So that is about the proteins and amino acids. Now we want to know about enzymes. Now we will come to know about the another important biomolecule, which is nothing but enzymes. So these enzymes are said to be the biological catalyst produced by the living cells. Okay, these are the enzymes and they are said to be the biological catalyst produced by the living cells. So these biological catalyst can catalyze several types of biological reactions in the living system. They are highly specific in their action. All the enzymes are conjugated proteins. All these enzymes are said to be examples of conjugated proteins. We have seen just now the classification of proteins, simple proteins, conjugated proteins and derived proteins. Okay, all these enzymes are coming under conjugated protein classification. So the pH and the temperature should be proper for an enzyme. Okay, each and every enzyme requires certain pH and temperature to be in active state, so to catalyze a chemical reaction or biochemical reaction. Here we will see few important types of enzymes which can which can act as a catalyst for different types of reactions. So first we will take enzymes this side and the reactions catalyzed by them. Reactions catalyzed by them. Okay, first I am taking here zymase. See, whatever the thing which is ending with the suffix ase, that is an example of enzyme. Okay, this zymase is used in the fermentation of glucose or fructose to ethyl alcohol. So for fermentation of glucose or fructose to ethyl alcohol. For this process, whatever the enzyme we are taking, that is nothing but zymase. Next one, invertase. Invertase is also one of the biocatalyst, or we can also call it as enzyme, which can catalyze sucrose into glucose and fructose. Sucrose into glucose and fructose. For this conversion, we can use this invertase enzyme. Next one, maltase. So this maltase can convert maltose into glucose and glucose. Maltose is one of the type of carbohydrate, that is disaccharide, which on catalysis by the enzyme maltase, it can be converted into glucose and glucose, because it is made up of two glucomeric units. So that maltase can catalyze the reaction. Next one, lactase. So this lactase can catalyze lactose into glucose and galactose. Lactose into glucose and galactose. Okay, this is also a disaccharide which is containing glucose and galactose monosaccharides. Next one, amylase. Okay, directly I will write the, okay, we will take amylase. So this amylase can catalyze starch into glucose. This starch is also known as amylum. So the starch is catalyzed by amylase to convert into glucose. It is a polysaccharide which contains n number of glucose units or glucose monosaccharide units. Next one, urease. So this urease can convert urea into ammonia and carbon dioxide. Urea formula is NH2CO NH2. So this urea can be catalyzed by urease and converts into two important molecules, ammonia and carbon dioxide. Okay, this can be catalyzed by the enzyme urease. Next one, pepsin. This pepsin is an enzyme where proteins can be converted into alpha amino acids in the presence of peptide. Pepsin. Proteins can be converted into alpha amino acid because most of the proteins are made up of alpha amino acids only. Whenever they are catalyzed by the pepsin enzyme, they can be converted into n number of alpha amino acids. Next one, trypsin. Okay, this trypsin can catalyze proteins into alpha amino acid. Another alternate enzyme which can catalyze proteins into alpha amino acids is trypsin. Okay, next one, nuclease. Nuclease is the enzyme which can catalyze nucleic acids into nucleotides. There are two important types of nucleic acids, that is DNA and RNA, deoxyribonucleotide and ribonucleic acids. So these two are the nucleic acids. These two can be catalyzed into nucleotides in presence of nuclease enzyme. Nucleotides. Next, another enzyme, lipase. So this lipase is used in the conversion of oils and fats into glycerol and fatty acids. Okay, oils and fats can be converted into glycerol and fatty acids in presence of lipase enzyme. So that means different types of enzymes can have different types of biological activity. So all these enzymes can catalyze different types of biochemical reactions. That's why we can call them as biological catalyst. We can call them as biological catalyst. So now we will discuss another important biomolecule which is present in the living system, very important biomolecule, that is nothing but nucleic acids. So these are the prosthetic groups of nucleoproteins. We can call them as prosthetic groups of nucleoproteins. Nucleoproteins are a type of proteins which is said to be prosthetic groups of nucleoproteins are called nucleic acids. So these are said to be biopolymers made up of nucleotide units. When n number of nucleotides are combined together, okay, so then these are called as polynucleotides. If only one nucleotide is present, then okay. Whenever n number of nucleotides, n number means many. When n number of nucleotides are combined, then that is said to be polynucleotide. Such polynucleotide is called nucleic acid. Such polynucleotide is called as nucleic acids. Okay, so these are present in all living cells. They direct the synthesis of proteins and are responsible for the transfer of genetic information. The two important uses of these nucleic acids is they direct synthesis of protein as well as they can transfer genetic information. So both can be done by these nucleic acids, that is DNA and RNA. Okay, nucleic acids are mainly of two types. Nucleic acids are mainly classified into two types. That is, first one is called DNA and the other one is RNA. DNA and RNA. So DNA is called deoxyribonucleic acid. Deoxyribonucleic acid. RNA is called ribonucleic acid. Okay, so they contains ribose as the sugar units. They contains two deoxyribose as the sugar unit. That is the only difference between these two. So this is containing ribose as the sugar unit and this is containing deoxyribose as the sugar unit. So that's three important things which are present in the DNA or RNA. Whether it is DNA or RNA, it contains three constituents. First one is nitrogen bases, next sugars, next phosphate units. We will see structures of DNA and RNA separately. First we will take RNA, structure of RNA. Okay, we know the complete form of RNA, that is ribose nucleic acid. So that ribose nucleic acid is made up of three important constituents. First one is nitrogen bases. Okay, the nitrogen bases present here are of two types, purines and pyrimidines. Purine, next one pyrimidine. Purines are of two types, adenine and guanine. Pyrimidines are of three types, thymine, cytosine and uracil. Okay, so three types of pyrimidine nitrogen bases are there, thymine, cytosine and uracil. Two types of purine units are there, that is adenine and guanine. So that is about the nitrogen bases. And second constituent present in nucleic acid RNA is sugar. So the sugar moiety present in this is ribose sugar. Okay, ribose is the sugar which is present in RNA, deoxyribose is the sugar which is present in DNA. Now the next constituent is, and the third constituent is nothing but phosphate unit. Okay, the phosphate units present here is the same. Okay, that phosphate unit can combine different types of nucleotides. Now we want to know what are nucleotides. So the three important units present or constituent present in RNA is nitrogen bases, sugars and phosphates. First we want to know what are nucleotides. There are compounds of base, sugar and phosphate unit. Nitrogen base, sugar and phosphate. Base, sugar and phosphate unit are called nucleotides. Okay, this is containing all the three types, base, sugar and phosphate unit. When n number of these nucleotides are combined together, it can be converted into polynucleotide. Such polynucleotide is called nucleic acid. Okay, clear? What is the sugar present in RNA? That is ribose. Understand? And there are two types of nitrogen bases, that is purines and pyrimidines. Now we will see the flow chart how DNA and RNA are formed. So first, how nucleic acids are formed, whether it is RNA or DNA. RNA, DNA both are formed by using nucleotides. Okay, when n number of nucleotides combined, that is called polynucleotides. When such type of polynucleotides can be involved to form either RNA or DNA. This nucleotide is containing two important basic units. First one is nucleosides and phosphoric acid. Okay, so nucleotide is containing nitrogen base, sugar moiety and phosphate. Nucleoside contains base and sugar. Okay, when base and sugar is present, that is called nucleoside. When nucleoside is combined with phosphoric acid, converts into nucleotide. That means nucleotide is containing base, sugar and phosphoric acid, whereas nucleoside is containing only base and sugar. So that these bases, okay, this nucleoside is containing nitrogen base and sugar, that we will write here. Nitrogen base and pentose sugar. Pentose sugar. So the two types of nitrogen bases present in nucleosides are, first one purines, next one pyrimidines. Okay, there are two types of purines, adenine and guanine. There are three types of pyrimidines, cytosine, thymine and uracil. Okay, in the same manner there are two types of pentose sugars. First one is ribose, next one is deoxyribose. Deoxyribose is present in DNA, ribose sugar is present in RNA. Pyrimidines and purines are the nitrogen bases which are present in both DNA and RNA. When nitrogen bases and pentose sugar are combined, when nitrogen base and pentose sugar are combined, they leads to give nucleoside. When this nucleoside is joined by phosphoric acid, it can be converted into nucleotide. So such nucleotides, when n number combined, converts into nucleic acid. Nucleic acid is made up of n number of nucleotides. Nucleotide is made up of nucleoside and phosphoric acid. Nucleoside is containing nitrogen base and pentose sugar. Nitrogen bases are of two types, purines and pyrimidines. Pentose sugar is of two types, ribose and deoxyribose. Ribose present in RNA, deoxyribose is present in DNA. Now we will see the main important units present in DNA, constituents present in DNA. So DNA is containing pentose sugar as deoxyribose. Okay, and then it is containing the nitrogen bases as adenine, guanine, cytosine and thymine. These are the nitrogen bases, and it is containing phosphate unit or phosphoric unit. So these are the three important constituents present in DNA. If you take RNA is containing which sugar? Ribose sugar. And the nitrogen base units present here is adenine, guanine, cytosine and uracil. Along with that, one more unit present here is nothing but phosphate unit. So these are the different types of constituents present in DNA and RNA. Okay. So the complete structure of DNA was explained by the scientist Watson and Crick. So the replication of DNA can be taken place, and it is transcription can be done to convert RNA formation. Now DNA and RNA are said to be the two important nucleic acids. DNA stands for deoxynucleic acid. This is ribose nucleic acid. Deoxyribose nucleic acid, ribose nucleic acid, and these are the constituents which are present in DNA and RNA. So DNA is useful for the transfer of genetic information from one generation to another generation. RNA is useful for the synthesis of protein. So that is about the nucleic acids.